Today we are going to start chapter 1 introduction and some preliminary considerations of embryology. Embryology Embryology is study of development of an individual before birth. It is also known as prenatal. Prenatal means before birth. Now embryo. The term embryo is coined when the baby is about 2 months or the 8 weeks inside the mother womb, and fetus it is called when the baby inside the mother womb is 3rd month or the 9th week till the time of the birth, and after the birth it is called as infant up to 1 year. Now basic qualities of living organisms, that is protection, growth and prolongation of species, also known as reproduction. Now gonads and gametes. The male sex organs, also known as spermatozoa, are produced in male gonads, testes, and this process is known as spermatogenesis, whereas female gonads, ova, are produced in female gonads, ovaries, and the process is known as oogenesis, and both production of gonads and gametes together known as gametogenesis. Now development of human being. The first step of development of human being is prenatal development or before birth development. In that we have many stages like pre-implantation or pre-embryonic period, where it includes first and second weeks of intrauterine development. Here first we can see the fusion of male and gamete occurs, which is known as fertilization. Then single cell zygote is formed and the primitive germ layers are formed. Here only double layer are formed, that is ectoderm and endoderm only. There is no mesoderm present in this step. So let us see in detail. We'll see the steps of pre-implantation. The first step is fertilization, that is fusion of male and female gametes to form zygote. Then cleavage. Then zygote undergoes mitotic division, or also known as equational division, to form morula, which is a 16 cell stage. Now transportation of cleavage zygote. Here morula starts moving from fallopian tube to the uterus. Now then after that it further differentiates and forms blastocysts, and it is structural and functional specialization and reorganization of cells of cleavage zygote that becomes the blastocysts. Now implantation. This is a major process in the implantation, where the process of attachment of blastocysts to the uterine endometrium is called as implantation. Now specialization of primordial embryonic tissues. It is specialization of blastomeres to form embryonic structures like embryoblast, and supportive or nutritive structures known as trophoblasts, which give nutrition to the fetus to grow. Then differentiation of embryoblast will occur to form the primitive two-layered germ disc having endoderm and ectoderm. Then differentiation of trophoblasts into cytotrophoblast and syncytios trophoblast. That is these are two trophoblasts which help in giving nutrition to the fetus. Now here you can see the process. The first cell or the zygote is here first of all, then it divides into the 16 cell stage known as morula. Then it forms blastula, which is fluid filled and hollow mass structure. Then differentiation occurs to form blastocyst, which contains differentiated cells, and it gets further differentiates and get attached to the mother's body and form placenta, umbilical cord. And placenta is the connection between the mother and child, which provides nutrition. And here you can see the first morula is moving from the fallopian tube to the uterus of the mother to get attached to it. Now let us know about second step of development of human, that is embryonic period, and it extends from 3rd week to the 8th week. Here first is trilaminar germ disc differentiation occurs. It is formation of 3-layered germ disc with an appearance of mesoderm in between the ectoderm and endoderm. As we saw in differentiation of embryoblast before, in the first step, that is pre-implantation, in that we saw that the primitive two-layered germ disc is formed, that is having ectoderm and endoderm, but mesoderm is missing. Now here in the embryonic period, we can see the formation of mesoderm. Then early organogenesis. Here, formation of primordia of various organs like liver, kidney, heart is going to happen. Then formation of extra embryonic supportive organs and membranes like placenta, umbilical cord, amnion, allantois, etc. Now the third step of human development is fetal period. Here it extends from 9th week to the 9th month. The growth of fetus and in all dimensions occurs in this fetal period, and specialization of various body structures occurs in this fetal period. Now the second step of human development is postnatal period of development. It extends from birth to adulthood. Now we'll see different branches, terms which are related to the embryology. The first is neonatal period. The birth it is the period between the birth and 28 days after birth. Here organs like heart and lungs get adjusted to the external environment. Now here neonatology. Neonatology is study of medicine which take care of neonates. Then prenatology. It takes care of fetus and newborn from the 28th week of intrauterine life of 6th day of extrauterine life. And infancy, it extends from 1 month to 1 year. The child is called as infant. Now childhood, 2 years to 12 years of age is known as childhood, also called as pediatric age. Now here pediatrics and pediatrician. The medicine that deals with children and infant is known as pediatrics, and the pediatrician is a doctor which deals with this medicine. Then puberty, it takes place between 12 years to 16 years. Adolescence is between 16 years to 20 years, and adulthood extends from 21 years to 40 years. Middle age extends from 40 years to 60 years, and the old age extends from 60 to till death. Now ontology. Ontology is study of complete life cycle of an individual organism. Now phylogeny, evolutionary or history of a group of organisms is called as phylogeny. Ontology always follows phylogeny in the study. Now embryology. Here it is also known as developmental anatomy, the science that deals with the process and regulations in the parental growth and development of an organism or individual in female genital tract is known as embryology. Now there are many subdivisions of embryology. Now let us know them. The first is general anatomy, the study of development during pre-embryonic and embryonic periods. The first 8 weeks after fertilization is known as general anatomy. Then systematic embryology, the study of formation of primordia and their structural and early functional organization into various organ and systems of the body is known as systematic embryology. Then comparative embryology, the study of embryo in different species of animals with compared with human is known as compared embryology. Now the fourth embryology is experimental embryology, to understand the effects of drugs, environment and pollution around us, the external, and to conduct experiment to prevent harm to the human development inside the mother womb is known as experimental embryology. And here we have biochemical and molecular aspects in embryology. Here the chromosomes, genes, sequencing regulations are seen in the biochemical and molecular aspects in embryology. Then teratology, it is a branch of embryology that deals with abnormal embryonic and fetal development, that is like congenital abnormalities or birth defects, etc. Now here defects of in women. For example, therapeutic procedures are used when defects are seen in the woman. Therapeutic procedures for infertility or fertility related problems. If a woman cannot reproduce by natural method, then cloning and in vitro fertilization can be planned. Then stem cell therapy, the cells forming tissues in the embryo are called stem cells. If we see the defect of stem cells, these cells are then the woman cannot reproduce. So these cells are capable of treating certain diseases in postnatal life. Now basic processes in embryology. First process is known as growth, and here we have types of growth. The first is multiplicative. It is predominant type observed during prenatal period. It increases the cell number by mitotic divisions without increase in cell size. For example, we can see this in blastomeres. Then auxetic growth. It is seen in oocytes and certain neurons. The cell size increases due to increase in its cytoplasmic content. These oocytes are surrounded by follicular cells and layer neurons by satellite cells to provide the nutrition. Then the next type is accretionary. It increases accumulation of intracellular substances resulting in overall growth of structure. These increase length, for example, increase in length of bone and cartilage. Then oppositional. This is additional of new layers on the previously formed ones, usually seen in the edges, seen in ridge structures, and is responsible for the contours. For example, increase in width of bone by addition of lamella. Now differentiation is the second basic process in embryology. It is of three types. So it is of four types. The first is chemodifferentiation. It is invisible differentiation that takes place at molecular level. The substances producing this type of differentiation are called as organizers. Then histodifferentiation. It occurs at tissue level. Then organogenesis or organodifferentiation. This is at organ level and the organ, and it is the basis for the organ remodeling. Then the next one is functional differentiation. Here hemodynamic changes occurs in blood vessel, etc. Now let us see the second basic process in embryology, that is organizer. Here primary we have three organizers. The primary organizer, example blastophore or primitive streak, that indicates the differentiation of notochord, and secondary or intraembryonic mesoderm. Then secondary organizer, example like notochord acts as secondary organizer in the stimulating development of brain and spinal cord. Then tertiary organizer, for example neural tube is a tertiary organizer that induces segmentation of paraxial mesoderm into somites. Now the third basic process in embryology is stem cells. Stem cells are undifferentiated cells capable of giving rise to more cells of same type. Now they are of two types: embryonic cells, somatic cells. Embryonic cells are present during embryonic period, that is when we are in the mother womb. Then somatic cells are the ones they remain throughout the life, or they are also known as adult cells. Now these bases for the formation of a tissue and an organ in the body. They have the capability of self renewal and differentiation. Now we have five types of somatic cells or stem cells. Now totipotent cells can form embryonic and extra embryonic cells. They are seen in zygote, early blastomeres, etc. Then pluripotent cells, all cell types of embryonic body, but not that of the placenta and umbilical cord, show these pluripotent cells. They are seen in inner cell masses. Then multipotent cells, more than one category of cells, like cord blood cells, stem cells, hematopoietic stem cells, etc. Then oligopotent cells, only one category of cells can be differentiated, for example vascular stem cells, etc. Then unipotent, only one type of cells can be differentiated, like liver cells. So we have completed the first lesson of embryology. Thank you.